We're going to talk about mitosis, which is a form of asexual reproduction. Asexual reproduction is when we have one parent cell that gives rise to what we call two daughter cells, and each of these daughter cells that are produced are genetically the same as each other, or as the parent, the original. As opposed to sexual reproduction, where you need two parents to produce one offspring, and that one offspring has a mix of both parents' DNA, 50% from each parent. So we're going to talk about mitosis and the different phases. The first thing that I want to bring to your attention is that it goes through multiple steps. Okay, the first step is prophase. Metaphase is the second. Anaphase. And telophase, which will bring us to interphase. And interphase just means it's between cell divisions, that it's not dividing and it's going through its daily activities. Okay, so we're going to concentrate on these four. So prophase, you're going to have a cell. Here's your cell membrane. Here's your nucleus. You're going to have a centriole over here, right? Because centrioles are important for cell division. Inside the nucleus, we're going to have genetic information. Right? And it's going to look like this. Now, this is what a cell will look like in interphase. If we call the DNA in this form chromatin, when prophase begins, what's going to happen is this nuclear envelope is going to start to break down. The centrioles are going to start. The centrioles are going to start to migrate to opposite sides, and you're going to notice that the DNA is going to start to condense. Now, you cannot see chromatin with a light microscope. So let's see. Let me see how we could do this here. So we have this. The nucleus is going to start to break down. You're going to get a centriole here and a centriole here. And they're going to start producing something called spindle fibers. We're going to see what they're used for in a minute. All right, so these are spindle fibers. And the DNA on the inside is now going to start to condense where you'll be able to see it. So we'll just do, this organism is just going to have two chromosomes. We're going to, it actually in this form, it's called a chromatid, where they look like, where it's a single strand of DNA. So that's a chromatid. All right, by the time we get done with prophase, the nucleus is going to be gone. You're going to have your centrioles here, centriole here. Now, the DNA, the chromatid, is going to copy itself. So it's going to look, it originally looked like this, right, from up here. But what's going to happen is it's going to make a copy. And now it's going to look like an X. All right, this is now your chromosome, or they call it sister chromatids, because this part here, this V, is identical to this V. So the genetic material or information on here is exactly the same as the one on here. It's like having two notebooks or textbooks that are exactly the same, has the same information. Same thing here. So these two are exactly the same. What will happen now is those spindle fibers that are here are going to attach, attach, and attach, and attach there. So the centrioles are going to start pulling, and using the spindle fibers as a rope. Okay, and what will happen is, let's go to the other slide, go to the next slide. This is going to bring us to the next phase, where when the centrioles are pulling, the genetic material now will be lined up down the middle of the cell. and we call that metaphase. So here they are, they're pulling. Think of like a tug of war. They're pulling, and they're lined up down the middle. This is metaphase. Then the next step is they're going to pull so hard that these sister chromatids are going to break apart. And we're going to be here. Okay, and so I got to keep switching colors. So now if you could see what's happening here, you're getting genetic material on opposite sides of the cell. So if we were just to draw an imaginary line down the cell, each half is going to have exactly the same DNA. Okay, this DNA, the genetic information found on this chromatid is exactly the same as this chromatid. The information on this chromatid is exactly the same as this chromatid. This, when the cell looks like this or is in this phase, this is anaphase, which will bring us next to the last phase. I'll draw that right here. Oops. Erase that. Now, this will be telophase. Oh, sorry. Now I'm just going to do a clean slide. Sorry about that. So the next step is telophase. Now what's going to happen here is, you notice the site of the cell membrane is going to start to pinch in. The nucleus will start to form. The genetic material is going to expand again. And eventually, we're going to wind up, this will pinch in all the way and separate, and we'll wind up with two daughter cells that are genetically the same. Same exact information. And this is what we call, this is what telophase looks like. When this is done, now these two cells are in interphase. And then the process will start over again once the cells mature and reach the right stage of interphase. That'll go into prophase and start replicating everything. So what I want you to do is now look into how mitosis is different in plant cells. The steps are exactly the same, right? You get the same phases, but there's one difference, right? Because remember, centrioles are only found in animal cells, not plant cells. So tell me how that happens, okay?